Hello guys and welcome back to Accessory Academy. So today we're going to talk about DNA topoisomerases. Like I have mentioned what DNA topoisomerases is, but the mechanism is required, as point of your exam. Entrance exam point of view के हिसाब से this mechanism is really important to understand. Okay, so we have mentioned that there are two types of topoisomerases. Okay, so before we proceed, so there are two types. One is there and two is there. So topoisomerase 1 and then topoisomerase 2 is there. So topoisomerase 2 in bacteria is also known as gyrase. Okay. Okay, then we just started. Okay, we just started. This is our first slide only. So I was just saying that today's topic is all about DNA topoisomerases, and there are two types of topoisomerase 1 and 2, where topoisomerase 2 is also known as gyrase in case of prokaryotes. Okay, so these are the class of the enzymes which alter the supercoiling of double-stranded DNA. Now, इन डबल स्ट्रैंडेड डीएनए सुपरकॉइलिंग कैसा जो आपका ट्विस्ट हो जाता है जैसे हमने वो टेलीफोन कोड में हम देखते हैं ठीक है so they act by these topoisomerases they act by transiently cutting one or both strands of the DNA. Very simple. Topoisomerase 1 is going to cut your single stranded DNA. ठीक है और सिंपल है topoisomerase 2 is for your double stranded. It is easy to remember also. Now, the regulation of DNA supercoiling is essential to DNA transcription and replication when the DNA helix must unwind to permit the proper function of the enzymatic machinery involved in these processes. So, ये भी हमें पता है कि and this is important, right? चाहे आप transcription करो, चाहे आप replication करो, we don't want any supercoiled DNA over there. Otherwise, what will happen? The transcription or replication which is carried out by the proteins which are involved in that won't be able to proceed further. Okay, so they are helping in maintaining both the transcription as well as replication of your DNA. Now there's something over here that you guys need to know, that is about the linking number and about the RIT, and of course with this it comes the twist also. So guys, do you, are you aware with this concept of linking number, RIT and twist? Have you heard of it?
Yes, ma'am.
Okay, good. Like अगर नहीं भी मालूम है, so please go through the chapter 4 of Watson. Okay, I have mentioned in the summary series also, like linking number क्या है, RIT क्या है, twist क्या है, very basic concepts are there. Okay, so just go through if you haven't gone through these concepts. Okay, so linking number. So topoisomerase का function देखो क्या है. So when you say that it removes the supercoiling, it means it changes the linking number, and your linking number is equal to RIT plus twist. Okay? So topoisomerases, they change the linking number of a circularly wound double-stranded DNA, where linking number क्या है आपका total number of times one strand of DNA helix is linked with the other in a covalently closed circular molecule. So this is what we are talking about in the case of the circular DNA. Okay. Now change in linking number will cause change in the RIT. Now RIT क्या होता है आपका? Number of times the DNA helix is coiled about itself in 3D space. समझ रहे हैं क्या आपको? लिंकिंग नंबर क्या हो गया आपका? यहां से आपको पता चलेगा लिंकिंग नंबर. RIT क्या होता है? RIT is like sorry. Okay. I don't know how much successful I am. Okay. So ये समझ में आ रहा है? RIT क्या हुआ? Your DNA helix is coiled about itself. So DNA helix आपका एक के ऊपर कितना कॉइल हुआ है वो आपका RIT हो गया है. Is it clear? लिंकिंग नंबर क्या हुआ? नंबर ऑफ टाइम्स DNA helix. Okay, यहाँ पे I'll show the figure, then it might be more clear. थोड़ा सा आइडिया मिल रहा है RIT क्या है, ट्विस्ट क्या है, लिंकिंग नंबर क्या है. Okay, I'll show the figure, then it will become more clear. Okay, so this is advantages. Sorry. Variation in TRIS subsequently changes the state of the compaction of DNA molecule. Now, this DNA compaction is also important. I'm sure you must have studied in the agarose gel electrophoresis that if you have a linear DNA, if you have a supercoiled DNA, और if you have covalently closed circular DNA, तो इनमें से सबसे पहले कौन आएगा नीचे in the agarose? Okay, the question is clear to you guys? Okay, no problem. I'll come back to this. I'll show you the figure, then maybe you might be able to answer. Okay, just let us keep this as aside. Okay. Okay, so this is advantageous because it permits the DNA to be transiently and locally melted to permit the enzymes for the DNA replication and transcription. Okay, so this much is clear to us that आपका जो DNA topoisomerase है, it changes the linking number so that the replication and the transcription enzymes जो हैं वो काम कर सकें, ठीक है? So as I already mentioned, there are two types, 1 and 2. अब इसमें भी important points please remember. So these enzymes remove supercoils by breaking one of the two strands of the DNA. आपका DNA डबल हेलिक्स है, राइट? वो सिर्फ एक स्ट्रैंड को कट करेगा और इसको ओपन करके यहाँ पे रीजॉइन कर देगा. So what will happen? So instead of having जैसे कि 1, 2, 3 and 4, what will happen? ये सिर्फ आपको 2 करके दे देगा. So this is how you're changing the linking number. Okay. So as a result, enzyme change the linking number by 1 each time. ठीक है? Now, in this case, the phosphodiester backbone chains are broken simultaneously and the linking number changes by 2. So guys, please remember this slide. This is important. Okay. Now, यहाँ पे क्या हो रहा है? The phosphodiester backbone is broken. What does it mean? It means both the strands, the double strand, जो double stranded DNA है, पूरा तोड़ा जाएगा. So you have got double stranded DNA like this. Okay. Then you have got another one like this. तो ये क्या है, आपस में टैंगल हो रखे हैं. तो इसका कॉइलिंग हटाने के लिए व्हाट विल हैपन इज योर वन ऑफ द डबल स्ट्रैंडेड डीएनए यहाँ पे दोनों कट किए जाएंगे. ओके, एंड इट विल बी ब्रॉट ओवर हियर. कट करके इसको पार कर देंगे हियर. So this is how the super coiling will be removed. Guys, are you getting the idea? थोड़ा सा idea. See, I just want you guys to imagine before you see the pictures. I have got the pictures. Okay, don't worry. Okay, so you got rough idea. Okay. Okay. So let's talk about the topoisomerase 1. It is best characterized in E. coli. It is monomeric. It is encoded by the top A gene. Okay, so mechanism of action. This is important. The mechanism of catalysis involves formation of covalent residue between tyrosine residue and the phosphodiester backbone. So what is happening? So we know that in the case of topoisomerase 1, one strand of the DNA is being broken, right? तो कैसा होगा? This is like आपने तोड़ा और उसको पकड़ के रखा है, ठीक है? Now this is held together by the help of the tyrosine of your topoisomerase, ठीक है? Nucleophilic attack from the hydroxyl group of tyrosine to a phosphorus atom is going to create a phosphodiester link between the enzyme and the DNA generates a free pi dash hydroxyl group. Okay, please understand this. The other strand is held in place by binding non-covalently to the domain of the enzyme. तो एक क्या हुआ है? आपका covalently attach हुआ है, ठीक है, to the tyrosine molecule. और एक क्या है? आपका non-covalently attach है. Now the strand is resealed. ठीक है, अभी क्या किया मैंने यहाँ पे? I made a cut over here. ठीक है, इसको कट किया तो ये स्ट्रैंड को मैं इस साइड ले आई और ये स्ट्रैंड को मैं इस साइड ले आई. सो फाइनली व्हाट इज हैपनिंग? आई एम गेटिंग लाइक दिस. सो दिस हैज बीन रिमूव्ड. ओके, so one linking number has been removed. Are you getting the point? 1, 2, 3 था, तो 1 और 2 मिलेगा सिर्फ. You have got 1, 2 and 3. So linking number was 3 over here. But with the help of the topoisomerase, I have removed 1. Now I have got only 2 as linking number. Is this much clear? Yes, so one supercoil is being removed. Now please remember, topoisomerase 1 from E. coli, it acts on negative supercoil. Okay, and it can remove both negative and positive supercoil. अब negative and positive क्या हुआ? It is just the direction. अगर right handed है तो हमने इसको positive बोल दिया, अगर उसका हेलिक्स अगर लेफ्ट हैंडेड है तो हमने उसको नेगेटिव बोल दिया, सिंपल. Okay, now let's see the figure. So you have got over here the double helix. Sorry, look at the double helix over here. So what you see, you have got 3 over here. Okay, now binding of the topoisomerase 1. So it is coming and it is binding over there. It is making a cut. तो आपको क्या मिलेगा, एक OH फ्री हो जाएगा और दूसरे जो स्ट्रैंड है वहां पे उसका जो फॉस्फेट ग्रुप है उससे टायरोसिन अटैच हो जाएगा ऑफ द एंजाइम. ठीक है, so this is where your DNA and enzyme intermediate is formed. Now what is happening, जैसे आपने एक को रीसील किया तो आपने इसको ओपन अप किया एंड दूसरे साइड में रीसील किया. सो व्हाट इज हैपनिंग, जहां पे 3 इन्वॉल्व थे वहां पे भी 2 हो गए. ठीक है, सो फाइनली व्हाट इज हैपनिंग, you have removed from 3, it became 2. Can you see the difference in the number? Yeah. Now the intermediate which is formed between the DNA and enzyme वो कुछ ऐसा दिखता है. So you have got DNA chain का 5' तो 5' जो है वहां पे आपका टायरोसिन के साथ कोवलेंट बॉन्ड बनता है and this tyrosin is of the topoisomerase and दूसरे स्ट्रैंड का OH रहेगा. Then again once it opens up the supercoiling का open होता है तो again same आपका OH फिर से phosphate के साथ अटैक करेगा न्यूक्लियोफिलिक अटैक and then you will have a phosphoester bond formation. ठीक है, so this is how topoisomerase 1 is acting. Okay, now let's see over here अभी enzyme के साथ. So first क्या है, you can see there is a binding. Okay, उसने cut किया, you can see that over here it is being associated right with the protein. Then आपने ये green वाला strand देखो, like look at over here the green color strand. What is happening, this strand has been taken that side. Okay, this strand has been taken that side और उसके पीछे से आके you are resealing the red वाला strand. The strand entrapment, then religation you're doing at the behind of that green wala strand. Then you release. So what is happening during this procedure, from if you had 3, it is becoming 2 linking number wala aapka DNA. Is this much clear? The figure is clear how topoisomerase is acting?
Yes, ma'am. Yes, ma'am.
Okay, good. So simple. So you have got these many, हमने इसको cut किया and then you can see the red वाला हमने इस side reseal किया. So that's how you get the loose. So this is how you're going to lose the supercoiled DNA. Okay. Now coming to the topoisomerase 2. It's a very small topic. Okay. So we can discuss this. Now talking about the topoisomerase 2. So it is also known as DNA gyrase and the structure. You can see the DNA gyrase is actually a tetrameric protein. वहां पे क्या था, topoisomerase, it was monomeric protein. Okay, this is tetrameric protein. Now it has got 2 subunits A and 2 subunits of B. Now depending on the DNA substrates, they can change the positive supercoils to negative or increase the number of the negative supercoil by 2. Now this is something which you need to by heart. Okay. And of course, yes, you can remember also. So क्या हो रहा है? So topoisomerase 1 can act on positive and negative supercoil. Topoisomerase 2 क्या करता है? Positive supercoil को negative कर सकता है या negative supercoil का supercoiling है वो बढ़ा सकता है. It can increase by 2 unit. Okay. Getting the point? See, अगर आपके DNA में you have got पॉजिटिव सुपर कॉइलिंग, ठीक है, अगर मेरा टोपोइसोमेरेस 2 आके उसमें 2 नेगेटिव सुपर कॉइलिंग ऐड करेगा, सो व्हाट विल हैपन, इज इट मेकिंग योर डीएनए रिलैक्स और मोर टेंस? Simple math. I'll repeat the question again. So I have got a positive supercoiled DNA. Okay. So this topoisomerase 2 is acting by increasing the negative supercoil or introducing the negative supercoiling. What is the net result over here? Whether the DNA is relaxed or more tense. और या मोर सुपर कॉइल्ड, क्या है इन दोनों में से सिचुएशन?
Relaxed.
Yes, it is relaxed. आपके पास पॉजिटिव था, पॉजिटिव में आपने नेगेटिव किया, तो अभी क्या हो रहा है? जीरो, यानी कि कम हो रहा है, राइट? तो सुपर कॉइलिंग कम हुई है. Okay. If I had negative supercoiled DNA, so it can convert to the positive. अगर negative को मैं positive कर रही हूँ, it means what? I'm again relaxing the DNA. It means I'm helping in the replication or the transcription required by the cell. Okay. Now, topoisomerase to catalyze carnation. It's not carnation, it is catenation and decatenation. That is linking and unlinking two DNA template. It also introduces negative and positive supercoil at the oric and removes positive supercoil ahead of the replication. Okay, so this we have discussed in the DNA replication that topoisomerase, it's removing the supercoiling. Okay, so इससे आपको याद रखना है, जब भी replication होता है तो वहां पे positive supercoiling होती है, जिसको remove करने के लिए we have got topoisomerase 2. Let it be bacteria, let it be eukaryotes. Please remember. Okay.
Ma'am,
हाँ.
ma'am, can it change from negative to positive? This introduce, I mean, it converts into negative supercoils, you know, from positive.
क्या
So
क्या? If
can
you
it do
have
the
got
ulta?
हाँ, yes, topoisomerase can do both, right? It can change positive supercoils to negative, or it can increase the number of negative supercoil. If it can increase more number of negative, तो क्या होगा? वो total negative हो जाएगा, right? See, it
Ma'am,
introduces
what if
negative.
the DNA is negatively supercoiled?
So then it is going to introduce positive supercoiling.
That's what was my question. So can it introduce positive supercoiling?
Yes. So it is depending on the DNA substrate कि आपका DNA positively supercoiled है या negatively supercoiled है. Depending on that, the topoisomerase 2 is going to act. ठीक है? Okay. Now coming to its mechanism. So one more thing which was not mentioned in the previous slide was that in the case of topoisomerase 1, you don't need ATP. Okay. So here you need ATP. So energy is required in order to carry out the mechanism. So hydrolysis of ATP by the ATPase activity of gyrase, it causes conformational change. Initially, enzyme binds to one part of the DNA strand. It is called G segment, inducing conformational change in all the A and B subunits of your topoisomerase. Remember, topoisomerase 2 is made up of two subunits, A and B. So there is going to be a conformational change. Okay. After binding the ATP and the another strand T segment, so G segment हो गया, T segment हो गया. A series of reactions occur in which G segment is cut by A and A dash domain of the enzymes. Okay. Okay, so you have got A dash and B dash subunits of topoisomerase 2. ठीक है? तो जो strand को cut करना है, it is called your G segment. और जो strand को सिर्फ pass करोगे, it is your T segment. Okay? So G segment is cut by the A and A dash domains of the enzyme, and ends of the GDNA become coordinated into the tyrosine. Again, it is the tyrosine residues. Simultaneously, ATP binding domains move towards each other, transporting the T segment through the bridge. अभी क्या है, जैसे कि मैंने mention किया, just let us imagine that this is your one double stranded, okay, and this is your second double stranded. अभी क्या होगा? Let us say that this is T segment and this is your G segment. अभी क्या है? G segment को cut करना है. So I'm going to cut this. Okay. Both the strands, both the double strand is being cut. Now what I'm going to do is I'm going to bring these strands over here, this side. तो अभी क्या हो जाएगा? ये यहाँ आके reseal हो जाएंगे and जो आपका T segment है वो बाहर की तरफ release हो जाएगा. So if I had like this, okay, अगर suppose मेरा ये DNA ऐसा था, ये G एक्ट करेगा, ये T एक्ट करेगा, लेटर ऑन व्हाट विल आई हैव, कट होने के बाद ये ओपन अप हो जाएगा ऐसा. क्लियर? समझा?
Yes.
हाँ? Don't worry, I mean, I have got the figure in videos. I just want you guys also to imagine, okay? So the cut G segment is really sealed and T segment is released by the conformational chain that separates the domains and then reforms. So alternatively, the enzyme can proceed another cycle. So please remember, you have got the two domains. ठीक है, आपके पास A subunit and B subunit के हैं. It's a total tetramer protein. Please remember. जिसको आप कट करने वाले हो वो आपका जी सेगमेंट है, जो पास होगा वो आपका टी सेगमेंट है, ठीक है। नाउ लेट्स सी द फिगर. Okay, look at over here. So you have got over here the figure. So आप देख सकते हो दो कलर में दिया हुआ है because you have got two different subunits over here. Okay, now look at, see, can you see it? So you have got this one as a T segment जिसको आप सिर्फ पास करोगे, जो रेड वाला है वो आप उसको कट करोगे. You can see that यहाँ पे हमने डबल स्ट्रैंड ब्रेक किया. Can you see it? There is nothing. It's a cut, right? इस कट से आप ये वाला जो है उसको पास करोगे. So you have passed the segment. Now at the back of this you are going to reseal it. Clear?
Yes, ma'am.
Huh? Now,
Yes, ma'am.
yes. So now you have got different types of conditions or different types of supercoiling where you can see. If suppose you have got like this tangled double stranded DNA, then you can see it over here. Cut kiya, reseal kiya, and you can have the separate double stranded DNA. Okay, or you can have like this also. पहले आप क्या देखोगे कि green ऊपर है, ये नीचे है. You can make it cut and again you can add it. So you can get the same thing. Green ऊपर होना चाहिए था, ये नीचे होना चाहिए था, वो भी मिल सकता है आपको, ठीक है. Now I think I have got one more figure which is more clear. So this is how it happens in the type 1. So एक strand break होता है और एक strand यहाँ पे आ जाएगा. Whereas in the case of type 2, both strands are broken, another double-stranded DNA is passed through and again reseal it. Okay. Yeah, so I have got the video. Let's watch this video. It will be more clear to you. 1 second. I think I need sound also.
Let's consider what
Are you able to hear the volume?
Yes, ma'am.
happens as DNA unwinds during replication. As DNA unwinds, it acts like this rope when we pull apart its two strands. As you pull the strands apart, twisting tension builds up in the rest of the coiled portion. It is actually adding one twist to the remaining rope for each twist pulled out of it. At some point, you can't separate the strands anymore. The remaining rope is too tightly twisted. If you relax your tension on the rope, it will twist around itself in a supercoil. It is releasing tension. If you want to keep pulling the rope apart, you have to release the tension periodically, and one way to do this is to cut the rope and splice it back together. This problem has been best characterized in small circular DNAs. There are two methods of dealing with this problem in DNA. One cuts only one strand of the DNA double helix, and the other cuts both strands. Let's look at the first. Topoisomerase I enzymes cut a single strand of the double helix, pass the other strand through the cut, and reseal the break, relaxing the overwound molecule, which now has one fewer twist. Topoisomerase II enzymes do the same thing but with both strands of the double helix. Topoisomerase II cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseals the break. So if a molecule of DNA is supercoiled, topoisomerase II can remove the supercoiling, 2 twists at a time, to yield a relaxed circle.
Okay, so guys, did you get the idea like how topoisomerase 1 and 2 are acting? Do you understand or do you want to watch the video again?
Yes,
वीडियो दोबारा देखना है या आगे बढ़े? Yes? Should I play the video again?
No, ma'am.
Okay, तो हम आगे बढ़ते हैं फिर.
Let's consider
Okay, so that was about the topoisomerase 1 and 2 कैसे काम करते हैं, ठीक है? The only thing is, like if you're interested, just please remember about the subunits and कैसे उसने काम किया है, ठीक है? And that part where we are talking about the G segment and T segment, That is also important. So you have seen कैसे उसने पहला जो है उसको होल्ड करके कट किया, right? So that is your G segment. जो पास हुआ वो आपका T segment. बस इतना याद रखना है. Okay. So then you can do many exercises which is there. Okay. So now inhibitors of topoisomerase. This actually I have taken in the previous lecture also where I have shown the table like जहाँ पे mention किया था कि gyrase को क्या inhibit करता है. Replication या I don't remember कौन से दिए थे, but I have taken this, but it's okay. We are taking it again. So they are the essential enzymes. Mutations to any of these gene coding for topoisomerase are usually lethal. Okay. See, it is lethal. It means what? Your cell is they are going to die. Okay. It's lethal means cell is gonna die. So therefore, they are the targets for the antibiotics. So अगर आपके पास if you are eukaryotic, we are mammals, right? What if, I mean, we are, yes. So of course, अगर antibiotics मुझे use करना है and I'm targeting prokaryotes, so what will I do? I can also target gyrase, है ना? So that they are not able to relax their DNA and the cell can die. Because जब आपका DNA relax नहीं होगा, so क्या होगा? The possibility of carrying out replication, the possibility of carrying out transcription, this will not be possible. जिसकी वजह से क्या होगा? The cell will not be able to divide. So this is how you can kill the bacteria. Okay. So they can be killed by novobiosin, nalidixic acid, the same thing which we have tabulated in the previous lecture. So they are the inhibitor of the DNA gyrase. So how does it doing? The mechanism behind the inhibition is novobiosin blocks the ATP binding and nalidixic acid blocks the breakage and rewinding mechanism. Please remember, novobiosin का कैसे तरीका है? So we have said the gyrase is like your topoisomerase 2. So it has got ATP binding domain. So you're blocking that ATP binding. So without ATP, it cannot relax the DNA. Whereas the nalidixic acid is blocking the mechanism of breaking and rewinding. Okay. Now, these antibiotics do not inhibit the eukaryotic topoisomerase. Of course, otherwise there is no point of using this antibiotics. We won't kill ourselves in order to kill the bacteria, right? Now, eukaryotic topoisomerases such as doxorubicin and etococytes are used as chemotherapeutic agents. Now, when you say chemotherapeutic agents, what does it mean? हम क्या target कर रहे हैं?
Cancer cells.
Yeah,
Cancer.
yes, good. So we are targeting cancer cells, but again, you should also understand that अगर हम cancer cells target कर रहे हैं, so there are chances that the normal cells are also targeted, है ना? Yeah, so that's why it is not a good way of giving the treatment. Okay. Now there is one way you can do. अगर आपका cancer जो है, it's not cancer. See, cancer when you say it means it has got metastasis. Okay. If you say it's a benign and if it is a tumor which is localized to one area, अगर आप सिर्फ उसको target कर सकते हो, then it is best way to treat the cancer. ठीक है? But basically, again, the best option will be what? To surgically remove the tumor so that it doesn't spread from benign, it doesn't metastasize, malignant नहीं बनना चाहिए. Correct? But yeah, there are so many, what to say, inhibitions, barriers are there. What if it is a brain tumor? Every time a surgical removal from the brain is not that successful. क्यों? Because a minor mistake, कोई भी एक neuron touch हो गया by mistake. Okay? If the chances of person maybe becoming the I say mentally paralyzed or having the dementia, ये सब फिर possibilities हैं. So आपको cancer से तो मुक्ति मिल गई, but the chances are that you are mentally impaired then, है ना? So that's why people are not going for the brain surgery if suppose you have got the tumor. It's like so many cases are there. Again, it depends which part of the brain the tumor is present. जैसे कि अगर it is local एक exterior portion में है, then it's fine. But just imagine if it is near the brainstem, then it becomes difficult. Okay. Oh. Okay. So let's do a few questions. The guys, you need to join me back because time is getting over. So like very few questions which I was able to get. I have put. Okay, so let's do this. So your first question is, action of topoisomerase leads to changes in dash. Yes. linking number of single-stranded DNA, linking number of double-stranded linear DNA, linking number of closed circular single-stranded or closed circular double-stranded DNA. क्या होगा आंसर? ऑलरेडी फिगर में दिखाया है मैंने. Yes, Akshaya?
Ma'am, I'm confused whether it is closed circular or linear, but it is double-stranded DNA.
Okay. So the figure where have shown, like in the video also, what kind of figure they have shown?
Closed circular.
Yes. Now what will be your answer then?
D.
Yes. Yes.
Yes,
Good.
ma'am. D.
So it is your D, linking number of closed circular double-stranded DNA. So obviously, please, this was very good. Like when Akhya said that just the confusion was between the linear and the circular. Obviously, the linking number will not come if it is a single-stranded DNA. ठीक है, वहां पे हम linking number, ये twisting, ये bridge का हम सवाल ही नहीं उठाएंगे. ठीक है. Only in the case of double-stranded DNA we are going to talk about it. Now linear stranded में क्या होता है? Sorry, linear. हाँ.
Ma'am, I'm not able to see the slide.
Who? Tanvi?
Yes, ma'am.
Okay, 1 second. What about others? Yeah, I guess they can see.
Ma'am, I can see.
Yes, ma'am. It's okay. It's fine now.
Now you can see Tanvi?
Yes, ma'am.
Okay, maybe आपकी तरफ से कुछ होगा issue, but now it's okay, right? Okay. हाँ, so I was explaining that question सिर्फ आपका linear and circular में आएगा. Linear DNA में कैसा है? Only like if you see the positive supercoiling is getting increased जैसे जैसे आपका replication होता है. ठीक है? Now only thing is that just imagine अगर suppose मेरा छोटा सा ही DNA होता, मैं इसको यहाँ से भी replication हो रहा है मेरा और यहाँ से भी हो रहा है. Just imagine, suppose I had small linear double-stranded DNA for replication. Do you think that the topoisomerase की जरूरत थी हमें? यहाँ से भी open है, यहाँ से भी open है. नहीं ना? Since we have got such a long stretch of DNA and there are multiple origin of replication, this problem is coming. Okay, but yeah, of course, we have got the topoisomerase to work. Okay, good. Now, next question. So, as topoisomerases play an important role during the replication, a large number of anti-cancer drugs have been developed that inhibit the activity of these enzymes. Which of the following statement is not true about topoisomerase as potential anti-cancer drug target? Okay, क्या होगा answer? Please देखो, हमारे टाइम पे नॉट को कैपिटलाइज नहीं करते थे. So many times we were like, you know, instead of giving the not ka answer, we used to give the opposite answer. So just consider this not true, okay, which is not true about topoisomerase as potential anti-cancer drug. Now tell me what will be the correct options. So your options are, as cancer cells are rapidly growing cells, they usually contain higher level of topoisomerases. The transient DNA breaks created by topoisomerase are usually converted to permanent breaks in the genome in the presence of topoisomerase targeted drugs. As cancer cells often have impaired DNA repair pathways, they are more susceptible towards the topoisomerase targeted drugs. The drugs which specifically target topoisomerase usually do not affect normal fast growing cells.
D.
Sure? D? Everyone?
Yeah.
क्यों?
Because they target normal cells also, because isomerases are present.
Very good, very good. So just now we had discussed that it's not like वो सिर्फ आपके कैंसर सेल को ही करेगा. It is also going to affect the normal fast growing cells. ठीक है? Good. So the answer is D. Good. So this was in your CSI 2016 first June में था शायद ये. Okay. So topoisomerase is also an enzyme which is used for carrying out ligation. The correct statement for topoisomerase is dash.
B.
Hmm?
B.
Okay, the problem with me is that for me, B and D sound same. Bombay B or Delhi D?
Bombay.
Okay. So the correct answer is they alter the degree of supercoiling of DNA molecules. Very good. See, topoisomerase is also for ligation. Why? Because first it is breaking the strand and then it is ligating. They act only on double stranded? नहीं. क्यों? क्योंकि topoisomerase सिर्फ बोला है, mention नहीं किया कि वो 1 है या 2 है. So that's why you cannot take this. ठीक है? Then they are less effective than conventional DNA ligase. ऐसा कुछ नहीं है. 3 types of topoisomerase ऐसा भी नहीं है. You have got many types of topoisomerase. If you see the classification, 1 के अंदर भी बहुत सारे type 1A, type 1-2 आएंगे. फिर topoisomerase 2 में भी आपको topoisomerase 2A, 2B ऐसे आएंगे. ठीक है? But since it is not required, I have never seen any questions like that, so I have not taken also. जितना है काम का, I have taken that. Okay, next question. Which special enzyme introduces negative supercoils in DNA?
MC.
Yes, very good. The correct answer is gyrase. Okay. So I have given the explanation also over here. It's a topoisomerase 2, which is found in the prokaryotes. It also introduces negative supercoil. Good. I think this is the last question. Now, attack of the topoisomerase gives rise to dash. Again, very simple.
B. Ma'am.
Yes, the answer is tyrosine wala bola na? Phosphotyrosine linkage.
Yes, ma'am. B.
Yes,
Yes,
ma'am.
B.
Yes. So explanation is also given and I have shown in the figure also. Okay, whether it is topoisomerase 1 or 2, both are going to have the tyrosine linkage. Okay, of course the phosphotyrosine linkage and आपका OH जो रहता है वो free रहता है. ठीक है, then OH is able to attack the nucleophilic attack, do on the phosphate group so that it can again form the phosphodiester bond back. Okay, yeah. So that's all, guys. I think we are done with the DNA replication. Any doubts? Okay, so far so good.